Good.
Okay. All right, everyone, we're going to go ahead and get started. Today's speaker is Hasan, and he's in the Mortazavi lab here. And he's going to be talking about today his pipeline that he's developed to analyze alternative polyadenylation for multiple different data modalities. So go ahead and take it away.
Hey everybody, I am Hasan. Today I will present LAPA, which is software we developed to analyze alternative pollination from long reads and short reads. First of all, why we want to study alternative pollination, why we care about alternative pollination, and what is alternative pollination first of all. So RNA molecules have a poly tail at the end, which is 50 to 100 base pair A's, consists of 50 to 100 base pair of A's at the end of the mRNA molecule. And this tail, like where the location of this tail or where the where is the alternate pollination site is important because it defines the 3' UTR region of mRNA. I will show you in the next slide, which is very important for regulation of mRNAs. So 70% of the transcripts have more than one poly site. And the poly site, the alternate pollination is very important for cell identity and tissue development. Moreover, this 3' UTR region defined by alternate pollination expanded during the evolution. As you see here, in C. elegans, this 3' UTR region is 140 base pair, and human is like 1-2 kb long. So this basically shows there is a complexity here, increasing complexity here, and misregulation of alternate pollination can cause a disease and cancer. That's why we want to study alternate pollination. So basically, you see an mRNA molecule over here, and as you see, this mRNA molecule can be terminated in multiple locations called alternate pollination sites. And each, depending on where it is terminated, you get a poly tail, as you see here. So we can get very long isoforms with long 3' UTR regions, as you see here. This red region, red region is the 3' UTR region. We can get very long 3' UTR region and following by poly tail. Poly tail is always there. Or we can get short 3' UTR region, or even we sometimes have alternate pollination sites in the exons or introns, then we can get different isoforms. And why this 3' UTR region is important is you can think it is a metadata. So if mRNA is a file, this region is the metadata of the file telling how to process this mRNA. And depending on this 3' UTR region, the macroRNAs, RNA binding proteins binds there and regulates this molecule. So this alternate pollination site, knowing the alternate pollination site is very important to know the transcript isoform and to know how this transcript isoform will be regulated. As I mentioned, if the genetic variants can disturb this alternate pollination process, for example, let's say the genetic variant in 3' UTR region can change alternate pollination, can affect our macroRNA binding. Similarly, 3' shortening and elongation can affect macroRNA binding. Let's say if it's a short isoform, macroRNAs cannot bind there. If it's a long isoform, macroRNAs can bind there. As a result of this post-transcript regulation, we get different level of gene expression, different level of protein like translation, and different level of protein. So all these post-transcript regulation are regulated by 3' UTR length, 3' UTR choice, and alternate pollination. So that's why we care about alternate pollination. So how to detect this alternate pollination site? How to know where is the mRNAs are terminated and where the poly tails appended? So there is different methods. The one of the methods is 3' sequencing. It's widely used method which only sequence reads with tails. So there is a specific adapter which enrich for poly tails, and the only regions where poly tails exist can sequence in 3' sequencing. It is a very specific protocol, just sequence pollination sites. Then we know precisely where is the pollination sites. The problem with this is a very specific protocol that is not widely available for all the data sets we have. And another issue, there is so many different 3' sequencing protocols. There is over 15 and there is no standardization. But there is no software to process 3' sequencing data. Everybody doing its own thing. They're writing custom scripts to process this data. Another data source to analyze alternate pollination, people tried, like again, there's like more than 5, almost 10 different tools to detect alternate pollination, is from short reads, the RNA sequencing we all know. And there, the major issue is that most of the reads in RNA sequencing on the middle. And there is very few reads at the start, at the end. That is because of something called coverage bias. That is the kind of the issue of short read RNA sequencing. The most of the reads contain in the middle, and there's little read at the start, at the end. So it's very difficult to know precisely where is the mRNA starting and where is the mRNA ending. And there's very few reads containing poly tail. So we don't know where the mRNA ends, where the gene ends almost like. And that is the major issue of short read RNA sequencing. So it's very difficult to detect alternate pollination. And most of the people trying to do this. The alternative approach is long reads. So long reads sequence complete mRNA isoform. It starts from the start of the mRNA, it goes until end, even it sequences the poly tails. So by looking at long reads, we can easily see where is the pollination site, where is the mRNA molecule stops and gets tail. So in this project, I developed a software called LAPA, which can process 3' sequencing regardless of the protocol and can process long reads to detect alternate pollination. So how we do this? Basically, we have reads with poly tails, either from 3' sequencing or from long reads. And then we detect coverage. We first of all count those reads with poly tail. We create the coverage tracks to understand what's the regions of alternate pollination. We do clustering, peak-calling annotation, and differential alternate pollination to study alternate pollination. So it is available in PyP. You can just download it. You can just pass your bound file, and either this 3' sequencing on long read, then you can pass your GTF file, and it generates you alternate pollination. So this easy tool is to use a single command. You can start studying alternate pollination from long reads or 3' sequence. So the first step, we look for poly tails if they exist, and then we count the pollination sites based on those reads. The next step, we do a clustering. We have a clustering algorithm which clusters those coverage and detect specific pollination sites. And furthermore, we need to define where is the exact location of pollination. For that, we need to do peak calling. And then we investigate where is those regions, is those alternate pollination regions in the exons, 3' UTRs. And another thing we look for, there's something called poly signal. So this motif always exists in the vicinity of alternate pollination sites. So we also look for this motif. So there should be this motif in the vicinity of our peaks. And another thing is a major issue when you study alternate pollination called internal priming. So there is some sketch of A base pairs in the DNA. They mimic poly tail. They are fake poly tails, and those need to be filtered. And we handle those as well. And at the end, we perform statistical testing. There is different testing options based on your needs. And we tell which genes have differential alternate pollinated. So those modules also have nice API. Those are defined with nice Python classes. So you can adapt those algorithms. Let's say you want to do peak clustering, you can use your data. We also write a nice Python API. So you can even use those steps, those components of LAPA for your specific purpose. Doesn't need to be like alternate pollination, but we have also a nice command line API, which you can study alternate pollination. So let's go to data, how the data looks like. So first of all, we have different protocols, different platforms over here. We have QuantSeq, which is a 3' sequencing library just designed to study alternate pollination. And there is a range of long read protocols. So long read RNA sequence is not as standardized as a short read RNA sequencing. In a short read, the Illumina is the most of the platform, and there is a couple of protocols, but they're highly standardized. So in the long reads, there's different options. There's two major platforms called TechBio and Nanopore, and there is different libraries, different ways to prepare your mRNA molecules. So first of all, we look how many reads generated based on each platform and how many reads contain poly tail or how many reads we can use to study alternate pollination. For QuantSeq, we have very deeply sequenced QuantSeq, which contain 90 million reads per replicates. As you see here, we have 3 replicates, and around 30 million of them have poly tails. So This is specifically designed for this library is designed to study alternate pollination. It's no surprise. There is high number of reads which can tell us the alternate pollination sites. If you compare long reads, as I said, the complete isoform. So by using long reads, almost all the reads mapped to genome can be used to study alternate pollination. And long reads still yield us millions of reads. And using these millions of reads, we can still study alternate pollination. It's not as specific as quansic, but still the numbers are comparable. It's in millions. The interesting part is the short-read RNA sequencing. Short-read RNA sequencing, we have 6 replicates, each have over 30 million, around 30 million reads. But if you subset for reads with poly tails, as I mentioned in my presentation before, the short-read RNA sequencing have coverage space. Only a few subset of the reads will have poly tail, and if you subset for those, you below 100,000 per replicate. So there is very few reads containing poly tail in the short-read RNA sequencing, which most people have try to study alternate pollination from short-read RNA sequencing, and it's not very feasible because most of the reads don't have poly tail. So, and if you compare those reads against the genome annotation, we still have millions of reads in the vicinity of annotated poly sites based on your GTF file, and we have very sharp peaks around annotated poly sites. So we can precisely study alternate pollination from long reads and long reads and quansic 3' sequencing. We didn't focus on the short reads because there's so many tools already there, and this data is not useful, in my opinion, not very useful to study alternate pollination. So we focus on 3' sequencing and long reads. So as I mentioned, this motif for AATAA is the canonical signal, and this signal needs to exist in the vicinity of alternate pollination sites. So we compare our alternate pollination sites. We first of all subset for the major alternate pollination sites for each gene we have, and we ask, do we have this signal, or what we are doing makes sense? If we have this signal, what we are doing makes sense. If you are missing the signal, something wrong with it. So we compare different protocols, for example, quansic and cDNA PEG-PAIR, those are the best ones. So for highly expressed genes, we have close to 100%, over 90% of our polycytes we detect have this canonical signal. So this figure basically shows us if we have enough coverage for those genes, which is TPM more than 5, we can precisely detect alternate pollination sites. Our sites we detect have this poly signal. And another issue is the internal priming. Internal priming is the major factor causing false positive poly sites. Our algorithm also handle those and filter those false sites due to internal priming. So the next thing we did is we compared how these different long read platforms and 3' sequencing compare with each other. Is there overlap between the poly sites we detect? So as you see over here, for most of the platforms, we have over 80% or close to 90% overlap between each other. There's high agreement. Of course, quansic detect many poly sites which cannot be detected by long reads because long reads are more shallow. The quansic is much deeper. So of course, there's some minor poly sites can be detected by quansic and not detected by long reads, but probably that will be solved in the future as long read depth increasing as technology evolves. So the next thing we did is we take quansic as a ground truth. So let's take the peaks we detected from quansic as a ground truth, and let's compare if we can predict those peaks with other long read platforms. And especially with direct RNA, CapTrap, we overall, regardless of the platform, we have very good performance. And there is some specific platforms such as direct RNA and cDNA have quite high performance. So we also tell the community which protocols is optimal to study alternate pollination. For example, if you want to study alternate pollination, and if we will do a long read, I think the best way to do is the direct RNA, because this protocol is specifically designed in a way that also detects poly sites with high accuracy. So we detect these poly sites and we also compare different platforms. The next thing we do with using long-read RNA sequencing, you can also create your own genome annotation. You can also create your own transcriptome annotation. And there is a tool we developed by Fairly, which called Talon, and Talon can create a transcriptome annotation from long reads. And I asked if with my tool, can I improve Talon? So Talon already have good performance. As you see here, for NNCs, like for there is different categories of transcripts, but overall, the many of the sites we have poly support. So we compare different protocols against the quantic data. As I said before, we get quantic 3' sequences with the ground truth, and we compare if the long read transcripts we detect from the transcripts we detect from long reads, the genome annotation we create from long reads have poly site support. And Talon have a relatively good performance, and using LAPA we can even improve this performance further. So as you see here, the transcript isoforms we detect from the different libraries, as you see here, and that is the baseline. So in the baseline, for example, for direct RNA, we have around 70% 3' support, and by correcting these ends further with LAPA, you can go even beyond 80%. So overall, we can even improve the genome annotation you created from Talon or any tool you have.
What is supported?
Supported by quantic 3' sequencing. So the transcript isoform is those transcript isoform supported by 3' sequencing. I will not go over there, but we can basically correct also star sites, and we can compare this against cache data. But let's go to more interesting stuff. Let's go to biology. So we have these myoblast cells and myotube cells. So there is a myoblast cells. Those are differentiated in the myotube cells with perturbation. So during this differentiation, we investigate, do we see a difference in alternative pollination? How alternative pollination signature change during this differentiation? And before we generated the long read data from those two cell lines, as you see here, we have two undifferentiated replicas and two differentiated replicas of myoblast cells and myotube cells. And then we run LAPA on there, and we investigate which genes are differentially alternative pollinated. So we detect around 720 genes significantly alternative pollinated between these two conditions. And we have an index of alternative pollination, which shows the site is pollinated or not pollinated between two conditions, as you see here, well, 0 to 1. And as you see here, between conditions, there is very clear alternative pollination signature. So alternative pollination is very important for cell identity, which we clearly see from this figure. There is many genes differentially alternative pollinated based on the cell state. So I plot those genes over here as a volcano plot, and the top genes we have before studied experimentally. So top genes we detect actually makes sense. There is a experiments like knockout experiments, knocking out those genes. Let's say I cite 4 different studies here. Each study knock out those genes, and differentiate do not happen if you knock out those genes. So those genes are extremely important for differentiation, that if you knock out those genes, differentiation do not happen. So the top genes we detect also make sense from this analysis. And another thing we see how 3' UTR length affects alternative pollination, how the gene expression, I'm sorry. So how 3' length affect gene expression, because 3' UTR length defined by alternative pollination. So depending on the alternative pollination site, you have different 3' lengths. So between the two conditions, we see that 3' elongation, longer 3' UTRs usually associated with lower expression, while shortening, if your UTR gets shorter, usually that is associated with higher expression. One explanation, one hypothesis we can have is there's macroRNAs targeting those transcripts, and depending on this macroRNA binding, we have different level of gene expression. MacroRNAs usually reduce gene expression. And when you have shorter 3' UTRs, macroRNA cannot bind because there is no binding site. Binding site is lost. And if you have longer isoforms, macroRNA can bind there. So that clearly shows that alternative pollination is very important for gene expression and it kind of defines gene expression in addition to cell state and cell identity. So overall, we develop a tool called LAPA, which can detect alternate pollination from long reads. It works with long reads and 3' sequencing. And overall, it shows that the differential alternate pollination analysis we have shows that alternate pollination is very important for cell state. And those post-transcriptional regulations are very important for cell state. And it defines kind of gene expression. Yeah. Thanks for listening. If you have any further questions, I can discuss.
Yeah, do we have any questions?
There's something in chat. So, yeah.
Okay, I have a question, which is if you so you basically had a test comparing the myotube and the myoblast to find alternative polyadenylation. What if you had like a couple more intermediate time points? Would you be able to find like how would you use LAPA to find like the polyadenylation that's like changing throughout the time course instead of just at like the start and the end?
Yeah, so the one place we can study this is the single cell, because during this differentiation, we also have single cell experiments. And if we adapt LAPA for single cell, then each cell, we can measure the ratio. And during the differentiation, we can like plot a nice UMAP, and then we can visualize for how each gene alternate pollinated throughout the time point, like pseudo time point at least. Yeah. And then we can see the signature, how signature change. Yeah, currently it's not adapted for short reads, but this is the one place we can improve, and we can even show like the time points.
But how would you do that like systematically, statistically? Like if you had more than two conditions, like more than just a pairwise comparison, you know?
So we implemented two statistical tests. One is the Fisher-Exec test, compare two conditions. If you have many conditions, and if you want to like detect outliers, let's say, you can use beta binomial test. We also implemented in LAPA. So yeah, depending on your needs, like we have two different statistical tests you can compare. Yeah.
Okay, so I had another question too. So I don't remember what slide it was on. So for, oh, it was for the heat map for the confusion matrix, the correlation plot. Slide 15, I think. Yeah, so you were talking about how like Quanseq's the gold standard and you think this is because of the read coverage. So I'm curious if you looked into whether or not the poly-A clusters detected in Quanseq but not in the long read were for lowly expressed genes.
Yeah, I haven't checked that, but that's a good question. I should investigate that. Like, but it doesn't necessarily lowly expressed genes. In the highly expressed genes, there could be also very minor poly sites, and Quanseq can also detect. Like, I think most of those is like there is in highly expressed genes there is poly sites used sub 1% or sub 5%, and Quanseq can detect those minor sites, and long reads cannot detect those minor sites because of the read coverage. So so this confusion matrix improves if you like subsets minor sites, it improves further. So I think some of them is unexpressed genes or lowly expressed genes, and some of them is the minor sites in the lowly expressed genes. Oh, sorry.
I was just wanting to ask about like where this project is. Like, are you wrapping it up right now? Is it already on bioRxiv? Is it already published?
Yeah, I'm trying to wrap up this project. So I like the, so it's tool is already publicly available, but the paper is not ready yet. So I have most of the figures as you see here. So I try to wrap up and try to put it bioarchive as soon as possible.
Yeah, it looks like it's like really polished already. So I was like, this looks like publication figures to me. So
Yeah.
Thanks.
Yeah,
Yeah.
basically, I prepared the figures. I need to write the paper. I'm working on the paper. I will like upload it to bioarchive as soon as possible.
okay, I also remember my other question, which was you tried it mostly on like cell line data. Does it look like, have you tried it on human samples or mouse samples and seeing like if the performance is better or worse?
So I haven't benchmarked the performance, but I guess the performance will be more or less same because there's no major difference in terms of biology or like in terms of sequencing. But I can benchmark in other human tissues as well. But the one thing we want to do, like we are working with Fairly, is we want to create like polya atlas because we have many human tissues and cell lines on the ENCODE. So by leveraging this data, we can create a polya atlas as well as transcript stat atlas, which will be like useful for wider community. Also, we can discuss further biology, not discussed in this paper about like human tissues.
So is that going into the ENCODE paper or is that its own, is that like something totally
This is
separate?
going to ENCODE paper.
Okay, cool.
In fairness.
Any more questions for the audience or from the audience? All right, if not, we can call it for this week's GenPALS. Just as a reminder, we're trying to do this office hours thing as well. So if you have genomics questions, we'll be in this same room at the same time next week. And we can either just work alongside one another or, talk about our projects and inspire one another, hopefully. So we hope to see you there. And until then, have a good rest of your week.